Hi everyone, this is Dr. Johansson giving you the first of several lectures for this class. I will tell you I'm a little new to, no not a little, very new to making these online lecture videos. So bear with me, I hope it's not too boring. But remember you can pause at any time and come back. Alright, so today we're going to cover chapters 2, 3, and 7, talking about bacteria, viruses, and prions. And the learning objectives for this week is really to get down the general characteristics of these different groups of pathogens and to think about why they are pathogens. So we're going to keep going into what do they do to the body, what are their characteristics that can cause us issues. So the first one we're going to talk about are bacteria, and hopefully you know that bacteria are prokaryotes, which means they have no nucleus, no organelles. The amazing thing about prokaryotes, though, is that they do cellular respiration. Some of them do photosynthesis. They do all the functions that eukaryotic cells do, just without compartmentalization. A couple other differences. The DNA is one circular double-stranded chromosome. Okay. They do have their own ribosomes, and the ribosomes are slightly different than eukaryotes. So this is actually a target for some antibiotics. So one of the things I want you to think about is when we're trying to develop drugs to treat these pathogen infections, we have to target something that's different than our cells because we don't want to take a medication that kills our own cells, right? So we look for differences. Another difference that is exploited by antibiotics is the cell wall, and we'll talk about that in a little bit. Bacteria also use slightly different proteins for DNA replication, so that can be a target as well. This little guy right here, which is called a plasmid, is a small circle of DNA, and it's self-replicating, and it's not part of the chromosome. So we'll talk about also that these plasmids are sometimes transferred from bacterial cell to bacterial cell, and the genes that are on the plasmid can give a new cell new genetic information, new characteristics. One of the things I want you to make sure you do when you're writing bacterial names is to always capitalize the genus, lowercase name the species. And then the entire name needs to either be underlined or in italics. Okay. Please, please, please write your bacterial names correctly. So for instance here, these guys really should be underlined or in italics. The other thing you want to make sure is that you don't start off with E. coli. Let's see if I can spell it right. Yeah. Hopefully I spelled that right. You always want to write out the formal name first, and then you can write out then you can use the abbreviation. Sorry, I'm just trying to check really quick and make sure I spelled. Oh. E. coli, right? E-S-C-H-E-R-I, sorry, C-H-I-A. Another thing that you'll see mentioned is SPP. That just means Shigella species. So that means multiple types of Shigella is what they're talking about. And then sometimes you see strain or serotype listed, and that gives you more specific details to the identification